All right, guys, I know some of you wanted to, sorry, I hear some noises. Some of you wanted to go over the CERR form and how you fill that out. So the lab title itself will be given to you either by me or if I'm not your teacher, then your teacher, or the lab itself, that lab packet. The research question is usually given there as well. But if you aren't sure, you can ask. Your null hypothesis is going to be where you're expecting the independent to not impact the dependent. So what you would be expecting here is that all of your groups would have the same results because there's nothing impacting them. So here you would say there will be no difference among the groups, or there will be no preference if you're looking at mice choosing food or crickets choosing smells or something. Alternative hypothesis, this is what you normally think of when you think of a hypothesis. So you would write your alternative, and it can be very generic. You could just say there will be a difference in the groups, or you can be specific and pick a group that you think will go higher or lower or whatever your values are. Your independent variable is what you are changing as the scientist. That would be if you're looking at different colors that you're shining on a plant to see if color affects growth. That would be the colors, because that's what you as the scientist are choosing. Dependent variable is your data that you're collecting. So that would be, in that example, that would be your plant growth. Your control group is the group that's going to be run pretty much as normal. So again, going back to the plant example, that would be with white light, because that's what plants are used to. They're getting the whole Roy G. Biv of the spectrum from white light. And then your experimental groups would be any other groups. So maybe you're growing plants in white light, blue light, and green light. This would be your blue and your green groups. Your data collection plan, you want to make sure you answer all three of these questions. What type of data are you collecting? That is almost always, I could probably say always, going to be quantitative data. We don't really work with qualitative in AP Bio. So you would just write quantitative over here. It does not need to be in complete sentences. What units of measurement? Put your units. If there's no units, write no units so that that question is still addressed. And then what basic equipment and procedure will you use? Keep it very short here, so like a sentence or two. And it doesn't have to be in complete sentences, but most of us write our procedures that way. So with our plants, you might say, I will grow 3 plants, 1 in white light, 1 in blue light, 1 in green light, or something like that. So very simple. Keep it simple there. And then your data analysis plan is what kind of statistics are you going to use? Are you doing standard error, standard deviation, chi-square? Those are really the only ones we do in AP. But if there was to be something else, then you would put that there. It does not need to be in complete sentences, so it could literally just say chi-square. You don't really need all these lines. All right, so that's the front of the CERR form, and you always want to do that before you start the lab. So once you get to this point, you would go off into the lab and do your lab, collect your data, make your data table, make your data chart, run your data analysis. So do your chi-square or your standard error or whatever. If you're doing standard error, you would be graphing those and adding your standard error bars to your graph and checking to see, because before you proceed, you need to know if you want to accept or reject your hypothesis. Okay, so you've done all that kind of in this space here, not literally in this space. You would do it on another sheet of paper and staple it to the lab, but in the time frame here. So now that you've done all that, you've collected your data, you've run your analysis, and you know whether or not you accept or reject your null, you come to the back of the CERR form and you circle either reject your null hypothesis if there is a difference, or if there's not, if your chi-square or your standard error bars show that there was no difference, no significant difference between the groups, then you would circle this, fail to reject, which means you accept your null hypothesis. Your claim, you're just going to restate your question as a statement. So if your question was, do plants grow better in blue light or green light, then here you would put, let's say that they do grow better in blue light and the ones in green light didn't grow and there was a statistical difference, then you would say here the plants grew better in the blue light. Okay, so you're just literally answering that question, the research question here. Evidence, do not put any statistics here. That's the biggest thing. This is literally just going to be your raw data. Whatever you put in your data table, you're just going to state it here as a sentence. So, and I'm just making up these numbers. The plants grown in white light grew 12 centimeters. The plants grew in blue light grew 18 centimeters, and the plants in green light grew 2 centimeters. Okay. So literally whatever your data is, you just put that here. This down here, how does the evidence support your claim? This is where you're going to talk about your statistics. So here you would say, I graphed my standard error, or I graphed my groups with my standard error bars, and the bars did not overlap, therefore I rejected my null. Or they did overlap, therefore I accepted my null. Or if it's chi-square, give that 4-part answer. Because the chi-square value was less than the critical value, and you would actually state those numbers. I just don't have numbers to state. So because the chi-square value of whatever your chi-square is less than the critical value of 5.99 or whatever your critical value is at p.05 and 2 degrees of freedom. So you talked about chi-square, critical, p-value, degrees of freedom. That's your 4 parts. Because the chi-square was less than the critical value, then I would accept my null. Or if it was more, then you'd say because my chi-square value of blah, blah, blah was more than my critical value of blah, blah, blah at p.05 and degrees of freedom, whatever that is, then I would reject my null. So that's all you have to put here, but that is where you want to keep all of your statistics. Somebody asked about mean. Mean can go up here because for our sake, mean is not a statistic. I know it is in math terms, but for our sakes, we're not using it to analyze our data. We have to take it a step further. So you can put means up here, but keep all of your standard deviation, standard error, chi-square down here. What scientific principle is involved? This one you're going to relate it back to to lecture. So this is how you're going to tie together your lecture that I teach you and the lab. So for the plant example, you would say, because plants have chlorophyll B, which uses blue light to do photosynthesis, this is why we know that the plants grew better at blue light than green light, because plants cannot use green light. Or something like that. So you would tie the lab back to what we've talked about in class. And then the easiest part probably is the reflection. What errors or flaws may have been present? Maybe the light wasn't shining directly on the plant, or something like that. And then where would you go next? I know for most of the experiments, you're not going anywhere next. You're done. But if you were, where would you go next? Would you test other colors of light? Would you test maybe different species of plant? Would you let them grow longer and see if the green plants ever catch up? Things like that. And so that is how you fill out your CERR form in a nutshell. If you have questions, let me know and we'll go over some more. I hope that helps.